Hi guys, welcome to Mr. Howe Biology. So this video is all about BTEC Applied Science and Forensic and Criminal Investigation, Unit 1, Cells. Now you know the Unit 1 exams include biology, chemistry and physics, and I'm going to help you to master these topics so that you get a distinction star in your final BTEC results. Now today's video is all about prokaryotic and eukaryotic cell structure. So what are the organelles you need to know about? What's their structure? What's their function? Well, today I'm going to cover all of that to help you get those top grades. So stay tuned, guys.
Okay guys, today we're covering BTEC Applied Science and Forensic and Criminal Investigation, Cell Structure and Function. So some key terms you need to know first of all. First of all, a prokaryote is a cell with no nucleus or membrane-bound organelles. And a classic example of this is a bacteria. Eukaryotes are cells with a true nucleus. And I always say to students, you do have a nucleus, as in eukaryote, you do have a nucleus. Pro, no nucleus. And eukaryotes include animal and plant cells. Now, organelles are small structures found within cells. So you can actually see in the diagram here, we've got the nucleus there. We've got the rough endoplasmic reticulum there. And we've got the Golgi apparatus there, just to show a few examples of organelles. Now, the ultrastructure is the structure within the cell, the internal structure within the cell. And we can see the ultrastructure of a bacterium at the top here and a eukaryotic animal cell there. So let's take a closer look at animal cell ultrastructure. You need to be able to label a eukaryotic animal cell for the Applied Science and Forensic and Criminal Investigation Unit 1 exam. So let's master this now. So first of all, we have the mitochondria. They have an inner folded membrane called the cristae and an outer membrane. They also have a fluid-filled matrix, and they're the site of aerobic respiration in cells, and they're where ATP is generated. So I'll just circle the mitochondria there. Now next up, we have the cytoplasm. Now this is the fluid-like substance, the gel-like substance throughout the cell, and it's where the metabolic reactions take place. Next, we have the lysosome. The lysosome is like a membrane bubble, okay? So it's a vesicle that contains digestive enzymes within it. And these enzymes are what we call hydrolytic, which means they break things down. So then we have the smooth endoplasmic reticulum. Now this is a series of flattened sacs called cisternae, and they synthesize lipids and carbohydrates. So those folds within them, they're the cisternae, okay? And remember the synthesis of lipids and carbs. Next, we have the ribosome. Now that's the site of protein synthesis. And because eukaryotes are larger and more complex, they also have larger ribosomes, and they're ATS ribosomes. Next, we have the centrioles. Now they're responsible for cell division, and they form spindle fibers during mitosis. We then have the rough endoplasmic reticulum, which you can see at the top right here. And this is made up of flattened sacs called cisternae, similarly to the smooth endoplasmic reticulum, but this time it's studded with ribosomes. And that's where it gets the name rough from. The ribosomes make it rough. And again, because ribosomes are the site of protein synthesis, proteins will be synthesized at the rough endoplasmic reticulum. Then we have the plasma membrane, which is also known as the phospholipid bilayer. And this is a selectively permeable layer that controls what enters and leaves the cell, as well as keeping everything together within it. Next, we have the nucleus, and the nucleus is this big organelle we can see in the middle, and it has a nucleolus within it, which is a chromatin-rich region. It has the nucleoplasm, which is the cytoplasm of the nucleus, and a nuclear membrane. So that's the special name given to the membrane that surrounds the nucleus. And basically, the DNA is located within the nucleus. It's a store of chromatin. And chromatin is a mixture of DNA and proteins. Now, the role of the nucleus really is to store the DNA, which is used to make RNA, and the RNA will later be used to make proteins. So next, we have the Golgi apparatus. Now, the Golgi apparatus modifies proteins with carbohydrate chains and forms transport vesicles. So let's have a look at eukaryotic plant cell structure next of all. Well, first we have the cytoplasm. This is the site of chemical reactions and a gel-like substance. We then have the nucleus, which again contains genetic material, very similar to eukaryotic animal cells here. We then have the cell membrane, which controls what enters and leaves the cell. Mitochondria, which are the site of aerobic respiration. And it's important that you know that plants respire too, it's not just animals. The vacuole, this larger organelle here, this is a key difference between plant and animal cells, and it stores cell sap, as well as maintaining turgor, which is almost kind of like the shape, the pressure within the cell, to support the cell. Next of all, we have the chloroplast, which is the green-looking organelle here, and this is the site of photosynthesis, and it's made up of stacks of membranes called thylakoid membranes, and they look like little stacks of pancakes if you look at them under the electron microscope, and they stack up to form grana. And within the chloroplast is chlorophyll, which is the pigment that absorbs light in photosynthesis. Next of all, we have the cell wall. Now, this is made up of cellulose, and it gives structure and support to the cell. We have the tonoplast, which is the cell membrane surrounding the vacuole. We have the amyloplast, which is a starch grain, and that has a double membrane. We have plasmodesmata. Now, these are channels between plant cells, and I've included a diagram here at the bottom where you can see two plant cells next to each other and the plasmodesmata connecting them. We have pits, which are pores in the xylem cell wall, and this allows water to enter and leave. Now, next of all, prokaryotic cell structure. I've got a bacterium here on the screen for you. Bacteria have a cell wall. Now, that's kind of similar to plant cells, but their cell wall is soft. That's because it's made of a peptidoglycan rather than cellulose. But the role here is still to support and protect the cell. Next, we have the capsule, which can often be referred to as the slime capsule, and this protects the bacteria against the immune response and prevents desiccation, which is the scientific word for drying out. So the slime capsule stops the bacteria from drying out. We have ribosomes within them, and remember, they have 70S ribosomes because they're smaller, and that again is the site of protein synthesis. We also have a nucleoid within the bacterium. Now, that's the genetic material, and it's circular, and it doesn't have a nuclear envelope around it. We then have the flagellum. Now, that's used for motility, meaning that the bacterium can move using the flagella. We also have the cell membrane, again, similar to animal and plant cells. This is going to control what enters and leaves. We have the pilus or pili, which is the plural, and these are going to allow bacteria to transfer genetic material. They're tubes that bacteria can connect to each other with and transfer their genetic material. Now, just a summary next of all. So eukaryotes and prokaryotes. So if we just run down the eukaryotic components that you need to know for your BTEC exam. Well, first of all, they have membrane-bound organelles. Eukaryotes have cellulose-based cell walls if they have a cell wall. They have DNA on linear chromosomes, so they're like matchsticks, it's not circular. The DNA is within the nucleus. ATP production occurs in mitochondria. They have a nucleus. They have large ATS ribosomes. They may have ungulopodia for movement. They have a phospholipid bilayer or the cell membrane. Now let's look at prokaryotes next. Well, they have no membrane-bound organelles. They have a peptidoglycan-based cell wall. The DNA is held on circular chromosomes. The DNA is contained within the cytoplasm. ATP production occurs in infolded regions of the cell surface membrane, and these are called mesosomes. They do not have a nucleus. They have small 70S ribosomes, which again are the site of protein synthesis. They may have flagella, and they have ribosomes, which are the site of protein synthesis. Now, I should have mentioned they also have a phospholipid bilayer too. Okay, guys, that's been the video today on prokaryotic and eukaryotic cells. I hope that's helped to prepare you for the exam. If it has, drop a like, comment and subscribe, and I will see you in the next one.